All right, so now we're going to talk about the reproduction basics of insects. Insects reproduce sexually, mostly. That doesn't mean there aren't asexual insects that are parthogenic. And most insects are oviparous, means that they lay eggs. The reproductive systems of insects are actually among the most morphologically complex parts of the insect. This complexity in their reproductive parts is a big part of speciation that prevents one beetle from breeding with another beetle, in addition to behavior and all sorts of things. Their reproductive parts are notorious for being quite ornate and structured differently and all sorts of things. They can be quite unique for species to species. The female reproductive system, and we'll go look at, of course, at pictures in a moment, consists of the ovaries, lateral oviducts and common oviduct, a genital chamber, a spermatheca, accessory glands, and ovipositor. In a lot of cases, insects can actually hold on to sperm. Females can hold on to sperm to allow their eggs to be fertilized at a later time than the reproductive act. So they can actually hold on to sperm and fertilize eggs over a period of time. Obviously, the accessory glands can provide nutrients and so forth. And the ovaries are obviously where the eggs are made. And then the ovipositor is where the eggs are laid. So these are examples of the female reproductive parts. You can see eggs and cells reproducing and becoming an egg that will then enter into the oviduct, and then from both sides will enter into a common oviduct, and then the eggs can be fertilized from sperm that can be held in the spermatheca, and then there's also a spermathecal gland associated with it. So again, females often can hold on to sperm for an indefinite period of time and then fertilize their eggs over a period of time. And then here's obviously where they would hook it up with the male and so forth. And then here's the accessory glands that are often providing nutrients and fluids and whatever else is needed in the reproductive system. Over here, we see the reproductive system of males. You got your testes, and then you'll see seminal vesicles, accessory glands, and then that'll lead to an ejaculatory duct and the beginning of the adiagus, which is obviously leading to the reproductive penis-like structure. Here is a female that is laying her eggs into wood. So this is the long ovipositor right here. So she can actually bore a hole. This is a type of wasp that can actually bore a hole into the wood and then lay her eggs into it. If you remember gall wasp that I showed you, they can also do things like this where they put their egg into a leaf, and then the gall is formed by the larvae feeding on it. This is a Jerusalem cricket, and it's going to lay its eggs, and of course, I'm going to lay it in the soil, or maybe on this leaf. But anyway, these are a bunch of eggs that have been glued together. So anyway, that's kind of the gist of female reproductive system. As I mentioned before, the male reproductive system consists of the testes. They're also going to have a vas deferens and a seminal vesicle, an ejaculatory duct, accessory glands, and the adiagus. where the hooks up with the female. And I think you can see that right here. The testes, then the tube is called the vas deferens. The mature sperm will come down to the ejaculatory duct, and then out the adiacus. And then here's the accessory glands. They're providing extra nutrients and solutions to the sperm. So, in some ways, even though they're very remotely, they're dramatically different than us as animals, you can see that some of the basic structures are there in insects as well as what we would see in us, the basics of it. So anyway, that's kind of the general gist of the insect reproductive system. They can do all sorts of interesting things to help find each other during mating season. As I mentioned before, males have very bushy antennae that help them to pick up on the female pheromones, which is like a hormone in a sense, but it's transmitted from one animal to the other, often through volatiles in the air. And that'll attract a male moth to the female moth that might be releasing the pheromones, for instance. In some cases, insects will do all sorts of things to kind of ensure their paternity, the males, where they may mate with the female and then guard the female so no other males can come around. They can often leave a sperm plug as well, where one insect will lay sperm into the female, and then they'll put a hardened plug on the outside of her reproductive parts, vagina, and so forth, so that another male can't penetrate and provide sperm. So there's all sorts of interesting ways that males try to ensure their paternity, as you can imagine, through evolutionary means. Selfish gene and all that kind of stuff that you may have learned in the class. Now let's talk about the sensory organs a little bit. This obviously is related to what we were talking about with neurons, but these are specialized cells that are associated with the neurons. And these include photoreceptors, and then mechanical receptors, which might be like little hairs, and chemoreceptors, which are little hairs that can pick up on the pheromones and smells of plants and so forth. Mechanical receptors means that it's going to be mechanically bent down and then send a signal to the nerve through the dendrites down through the axon. We call that an action potential, and then to cause muscles and things to contract. And then we have photoreceptors as well. So in a sense, in that picture with the robo cockroach, that metallic needle that kind of went down the antenna would stimulate the antenna, whether it be mechanical or some kind of stimuli that the insect brain would perceive. It could have been a mechanical or chemical receptor. Both of them may have been affected by that electrical signal that would cause the cockroach to move in different directions. Now, going back to photoreceptors, these are basically what is needed to pick up on light energy. So there's special pigments that are found on the eyes or on the ocilli. And if you're familiar, you have pigments in your eyes as well, like rhodopsin and retinol. There's a light absorption that takes place there in the retina. There's also different rods and cones that pick up on that light energy where the retina will be. And maybe you remember a little bit of this from an animal physiology class. Well, they have similar kind of things where the pigments will pick up on light energy, and then they'll convert that electromagnetic energy into chemical energy that causes nerves to depolarize or hyperpolarize, depending on the case of how it's set up, but ultimately initiating neurons that will then travel to the brains or the ganglion to cause the insect to move or respond to the environment or move to the light or move away from the light or whatever it's been set up to do, evolutionarily speaking. So the photoreceptors are going to be found in the compound eyes. Remember, there's multiple facets that make up the compound eyes, and there's going to be pigments found in those cells, or they're going to be found in the oscilli, which are, again, those little spots that you can find on insect heads. They can pick up on light. And apparently a structure called stomata as well. So the compound eye is in nearly all adults and some immature insects. So you don't see compound eyes, for instance, on a typical caterpillar, but you'll see the compound eye on the butterfly. And so they're composed of many ommatidia, which are basically the facets that join together to make a compound eye and the functional unit, and each one of those are the functional unit of vision. So they don't just have one eyeball, they actually have multiple eyes that come together to make a unit of vision. So each one of these is going to pick up on light. So they're going to have a lens on the outside. Light's going to travel through, hit pigments, and then cause the cell to respond, and then send information down neurons to the brain. So here is the ommatidia zoomed in and with labeling. You can see the corneal lens on the outside. And then you're going to see the pigment cells right underneath it. So the light will hit these pigment cells that will turn that electrical chemical stimuli into electrical magnetic to electrical chemical stimuli that will depolarize the cells, and then apparently they'll travel down these this rhabdom to the nerve cells down here. And then you have various support cells as well. And these include these pigment cells over here and these retinal cells. So the light energy comes through and will stimulate, I believe, these retinal cells. Let me double check on that. But here's the compound eye right here. You can see all the different facets. And so each one of these has a corneal lens on top. So you can see that. So their vision is going to look kind of multifaceted, but the brain will put together the image. So they're not really sure how the good insects can see. They have different ideas, like the mosaic theory, that each one of these eyes helps put together a picture, or that they may have individual pictures, and it's not really clear how good their resolution is. I mean, it's presumed that they're not seeing in HD, let's put it that way. But as you know, that they are effective for this housefly not to be swatted by you. So they are effective and probably mandatory for their body size. So they have superior motion detection, but poor resolution. Here are some examples of insect eyes. Here is the caterpillars or larvae, and you'll see that there's oscilla, and I guess together they make up the stomata, if I remember right. And then when you have multiple ones together, they're called stomata. And if they're just a couple on the top, that's dorsal ocellus. And then you have compound eyes again, that you can see very clearly on odonata. Hopefully by now you don't realize odonata, it means dragonflies. Or various types of coleoptera, which you should know by now is beetles. So hopefully you know those terms by now. And then various other types of insects like Neuroptera and Thysanoptera and so forth. And then Drosophila, which are part of Diptera. We should know that those are flies at this point. So the oscilli, for the most part, they're not providing any information as far as what the image looks like. It's mostly about light and dark. And so it allows the insect to respond to light intensity and helps them with their circadian rhythms and their daily behavioral rhythms. And then it might help them to move a little bit in regards to the horizon. So this is oscilli right here. So here's compound eyes, and then those little dots are the oscilli that help with light detection. And then stomata is like a circular group of oscilli, I believe. Let me double check that. But we can find this in holometabolous larvae, which means the larvae that can go through complete metamorphosis. And they have high light sensitivity, but again, they're low resolution, so they can't really see very well. So it's again providing information on how bright it is and moving from bright areas or dark areas or whatever the insect might need. And so that's what those stomata are right here. So they're like a bunch of oscillis joined together to make the stomata. Mechanical receptors are found almost anywhere on the insect body. Again, this is their way of detecting mechanical stimulus. They can be bent or stretched, compressed, and all of them basically are providing some information about touch to the insect. So some are tactile, but some are also proprioceptive, which are basically helping with body orientation. Like even on ourselves, you can tell where your hand is by the nerves that are in your hand, sending information to your brain. That's proprioceptive. Or if your stomach stretches or stretch nerves and things like that, that's all examples of proprioceptive that allows for your body to know something about its orientation or so forth. Then there are sound reception, and insects don't have classical ears, yet there's membranes that vibrate kind of like eardrums, and they can be found on the forearms and various regions of the insect's body. And again, they can respond to the sound waves that will hit it. So some of the mechanical receptors include trichiform sensili, which have these little hairs that are tactile that can bend, and then they'll send a signal down the nerve. So this is the hair, and if you were to touch it, the hair would bend, and then that would trigger either a release of neurotransmitters like it happens in us, or something that might bend the dendrites as well. But it'll cause the neuron to undergo an action potential. Remember, this is the cell body of a neuron. This is the nucleus. This is the dendrites, and then the information would travel down the axon to cause the muscles to contract or something of that nature. And so there's all sorts of different types of mechanical receptors. Some include campiform sensili, which are basically flex receptors. So they might be a little flatter and they can tell when the body's bending. And again, what happened is this hair will bend, send a chemical signal down the dendrites and then down the nerve cells. This is the nerve cell right here. And this is the campiform syncytium, which is kind of like a hair cell on top of it. So this would be the nucleus of the hair cell. That's what I'm calling it for the purposes of this class. And then they might have some connective tissue around it. And so when it bends or the body bends, they'll feel that flexion. And then again, some of the mechanical receptors are involved in hearing and sound and vibration. And these are known as cordonial organs. And so they can pick up on sound and vibration, and tympanium, which will be a membrane that can bounce with sound waves. And so those can be found on the grasshopper's leg, for instance. And then there'll be sound waves or whatever vibrations will hit those membranes, and they'll move and jiggle. And then they'll either bend hair cells that send chemicals to neurons, or bend the neurons themselves, but ultimately causing the neuron to depolarize, if you remember that term. If not, it might be good to check a book on neurons, or just watch a video on it. And again, I have a YouTube video where I talk about neurons. Now, chemoreceptors are basically picking up on the chemicals that are found in the environment. Some are known as olfactory receptors, so that obviously would be something that the insect smells. It's not going to be through a nose, but it could be found on hairs on their body, or on their antenna in some cases, or obviously in a lot of cases. Again, it's going to have a kind of a lock and key mechanism, where the chemical has a certain structure and it'll attach to the receptor that's found on the antenna or parts of the body. And then those cells release chemicals or neurotransmitters or something in some manner to the chemical that causes the neuron to release a signal that will travel to the ganglion or the brains or so forth. Taste receptors can be found on their feet as well as around their mouth parts. Again, it's picking up on chemicals, but these, in this case, the chemicals are in some type of liquid. Olfactory is obviously going to be found in gas, but the same idea, a chemical is going to attach to the receptor in such a way that it triggers that specialized cell to release neurotransmitters or not, but because either one of those things can be communication to the neuron, depending on whether it depolarizes the receptor or not. So again, olfactory receptors are chemicals detected as odors. They're gaseous, airborne, and usually in low concentrations. There's going to be many neurons associated with it if it's multipolarous. Most of it is abundant on the antenna or thin-walled pegs that are associated with different regions of their body or legs. And so this is the hair-like projection sticking out. So a chemical can come along and attach, triggering the hair cells to release chemicals that will then stimulate these neurons to send information to the brain. So those would be multiporous in that case. So this would be a multiporous chemoreceptor. Some people look at electrotenograms to figure out how the antenna respond to chemical stimuli. Our faculty member, Dr. Jeff Engel, has looked at a lot of chemical electrotenograms in regards to different kinds of responses. But what happens in some researchers, what they do is they'll take a chemical, maybe a perfume that they extracted from an apple, and they'll spray it over the insect's antenna, and they'll see that the antenna responds to it by giving an action potential that they can measure on a special electrotenogram device that has a needle that goes into the antenna, and then it provides evidence, or proof in quotes, but evidence that particular chemical the insect will respond to. So that's what an electrotenogram does. So again, this is usually combined with someone purifying a chemical. How do they know they purified it? They use or they give an extract of a chemical. They'll often do some chemistry stuff using a GC mass spec. That is gas chromatography, mass spectrum or spectrography. Anyway, tongue tied. But what it does is it's a way for a researcher to look at a peak of a chemical. So they sprayed this chemical into this machine. Maybe you know a little bit about it from taking a chemistry class. And the machine will show a peak, and then they'll look at the insect's antenna that's gotten the puff of that air. And they'll say, oh, look, this chemical methyl jasmate stimulated the antenna in some manner. So they know that the chemical the insect can detect. So that's how they figure those kind of things out. So they can look at it and get an idea of what the insect might be attracted to based on the insect's response to a particular chemical. So they can actually using chemistry, make some things more attractive to the insect that literally tracks the insect to a trap, like a pheromone trap. So pheromone traps is a good example. So have you ever seen trees where they have a box that's kind of triangular shape? Well, those things are releasing pheromones that pull insects into it, and then there's a sticky trap in it that'll catch it or whatever. But they figured that out by doing these electrotenograms where they had a needle in the insect's antenna, and then they give it a puff of some type of gas and see whether or not it responds. Again, taste receptors are found on thick hairs. Again, made up of the cuticle, it's not really a hair. Or they may be pits or pegs that are sticking out of the insect, or pits. And they can either be phagostimulatory, meaning that they'll increase the likelihood of the insect going to the chemical and wanting to eat it, or they could be a deterrent and actually cause the insect to run away or fly away or whatever the case might be. Like DEET can chase away mosquitoes. We learned that through using these tests and so forth. Again, taste receptors are found on the antenna, the mouth parts, as well as the tarsi and the ovipositor. Remember, the tarsi are found on the legs of the insect at the end of it, while the ovipositor is at the tail end. So the tail end will have chemical stimuli that can taste the leaf or whatever they're about to lay the egg in, or the insect, or whatever they happen to be laying it in. This is the hair cell. And so again, it'll have the ability to pick up chemicals like a lock and key, and the chemical will attach to the receptor, cause the hair cells to respond and send a signal down the neuron. Or there might be pits and so forth. Anyway, that kind of gives you a little bit more information about how insects respond to the environment. Let's take a moment and see if we can find an electrotenogram or any more information easily on YouTube for about 10 minutes or so, and then we'll call it a day, or 15 minutes or so. So that finishes up that lecture. And then, like I said, I encourage you to go to YouTube and explore more animation-filled presentations. Thank you for listening.